From the Library of Congress in Washington, D.C.
Good afternoon, I'm Fenella France, Chief of the Preservation Research and Testing Division, and I'd like to welcome you today to our TOPS lecture on medieval medicine for modern infections. Could ancient biotics help in the fight against antimicrobial resistance? I'd like to introduce our speaker today, Dr. Erin Connelly, who's the CLIR-Mellon Fellow for Data Curation and Medieval Studies in the Schoenberg Institute for Manuscript Studies and the Kislak Centre for Special Collections, Rare Books and Manuscripts. She holds a PhD in medieval English from the University of Nottingham, and her doctoral project was the first edition of the Middle English translation of Bernard of Gordon's Lilium Beniciniae, a significant medieval medical text. She also holds a BSc in biology and worked briefly in molecular oncology. She's part of the Ancient Biotics team, which explores the relevance of medieval medicines for modern infections. I know you're going to find this absolutely fascinating, and I'm going to hand over to Erin.
Thank you very much to Fenella and to everyone here at the Library of Congress for inviting me to speak today. I'm very much looking forward to sharing this research with you. So, we have a few objectives for today's talk. In summary, the Ancient Biotics team tests the efficacy of medieval medicines for modern infections. And I'm going to be talking about a successful pilot study on a 1,000-year-old recipe known as Bald's isolve, which turned out to be a potent anti-staphylococcal agent. It repeatedly killed Staph aureus biofilms in vitro, and it killed methicillin-resistant Staph aureus, or MRSA, in a mouse chronic wound model. Now, because this pilot study occurred in 2015, we'll also be talking about current and future research plans, of which there are many. Now, I'm going to be presenting scientific evidence from the pilot study, but I speak to you today as a medievalist. And although this research does hold implications for present-day drug discovery, it also raises very interesting questions about the methodology of medieval practitioners. And for a long time, the Middle Ages, medieval medicine, it has been dismissed as irrelevant, overly superstitious, a time of gathering information, solely to gather together information. We're all familiar with the term Dark Ages, labeling the period as unenlightened by science or reason. However, recent scholarship may show that there is more methodology to the medicines of medieval practitioners, and further inquiry may show that their medicines were more than just placebos or palliative aids, but actual antibiotics being used long before the advent of modern infection control. So as a medievalist, I thought it best to begin with a story from medical history. What you see on your screen is an excerpt from Thomas Gale's Certain Works of Surgery. Now, Thomas Gale was the 16th century master of the London Barber Surgeons Guild, and he is connected with the provenance of one of the texts studied by the Ancient Biotics team. Fenella mentioned it. It's known as the Lily of Medicines. In the folio preceding this excerpt, Thomas Gale relates a personal case study of a young boy with sore eyes. He says, "Every member of the guild is working on treating this child. They try all their treatments, but it only brings the child's condition to worse. Until Gail says, "We left the child to the work of nature." However, the child's mother found a recipe in an old book, brought this recipe to Gail, who distilled it, applied it to the child, and as you see underlined in the slide, he says, it restored the child's eyes to perfect health and sight. Now, although Gail's account is several centuries old, it is analogous to our current situation. At present, there is an urgent need for novel routes to drug discovery due to the emergence of antibiotic-resistant bacteria and the lack of new drugs currently in development to treat antibiotic-resistant infections. The World Health Organization stated in September 2016 that antimicrobial resistance is an increasingly serious threat to global public health that requires action across all government sectors and society. In 2017, the CDC related a case study of a woman in Nevada who died of an infection that was resistant to every known antimicrobial in the United States. Current estimates say that around 700,000 people a year around the world die of antimicrobial resistant infections. And if no action is taken, that estimate jumps to 10 million people by 2050. Now on your screen here is a dramatic gesture by Image Comics, funded by the Wellcome Trust, to capture broad public attention about this threat to health. This is a comic book series that envisions a post-antibiotic dystopia, actually in a much nearer future than 2050. This one is set in 2036, in which millions of people are dying of antibiotic-resistant infections. Like Thomas Gale's remedy from an old book, there are some historians and scientists who believe that the answer to the antibiotic crisis may be found in medical history in combination with modern technologies. So in response to this pressing global threat, the Ancient Biotics team was formed in 2014. It is a truly interdisciplinary and international team. We are united by the belief that novel routes to drug discovery are necessary, as well as the conviction that the past could inform the future. So along those lines, I'm going to present some of the evidence from the pilot study on an eye remedy called Bald's Eye Salve. And the following slides represent data taken from the original paper, which was published in InBio. It's available online, and it represents work done by Freya Harrison, Alid Roberts, Rebecca Gabrilska, Kendra Rumbaugh, Christina Lee, and Steve Diggle. So here it is, Bald's Eye Salve. This is the recipe as it appears in a book called Bald's Leech Book, 1,000-year-old remedy book held by the British Library. Now, it is written in Old English. This is an Anglo-Saxon recipe, but in modern English, as translated by Christina Lee, it reads something like, "Make an eye salve against a wen. Take equal amounts of an allium species and garlic. Pound them well together. Take equal amounts of wine and oxgall. Mix that with the alliums. Put this in a brass vessel. Let the mixture stand for 9 nights in the brass vessel. Wring through a cloth and clarify it well. Put in a horn. and at night apply it to the eye with a feather. This is the best medicine. Now, a win in modern terms could be translated as a stye, which is an infection of the eyelash follicle. And in modern terms, the most common cause of infection is the gram-positive bacterium Staphylococcus aureus, which also causes other severe and persistent infections. So what does the data show? This is what the eye salve looks like. And because the allium species in the recipe is a little bit ambiguous, a leek version and an onion version of the eye salve was made. And this is because leek and onion, they both contain antimicrobial flavonoids not found in garlic, which is already part of the recipe. And onion also contains an antimicrobial peptide. So 2 versions of the eye salve were made, one with leek, one with onion. Preparations of individual ingredients, versions of the recipes with ingredients deleted, and then preparations of the recipe to be tested before and after this 9-day activation period were also prepared. And it is noteworthy that after these 9 days, the recipe is self-sterilizing. So first round of tests. Bald's eye salve kills Staph aureus in planktonic culture and in a synthetic wound biofilm model. In figure A, a control of sterile distilled water, which is represented by the black line, as well as the 2 versions of the recipe, onion and leek. Onion is represented by the red line, and the leek is represented by the blue line, were added to Staph aureus, growing in synthetic wound fluid, and then the optical density of the culture was taken during 18 hours of incubation. You can very clearly see in this image the difference in activity between the control and the 2 eye salves. In figure B, a control, again, of sterile distilled water, the 2 variants of the eye salve, onion and leek, and then each individual ingredient on its own were added to Staph aureus, growing in a synthetic wound biofilm model. Here, the asterisks denote treatments that are significantly different from the control. And you'll note here that on their own, each individual ingredient had no significant effects on the reduction in bacteria. However, the highlighted version of the 2 full recipes did. And this is a key idea, this combination of ingredients. This is where we seem to see this potent anti-staphylococcal activity. So along those lines, the activity of Bald's eye salve against Staph aureus biofilms requires several ingredients. What is this image showing? Here, there are the 2 versions of the eye salve, the full recipe, onion and leek, and then variants of the recipe with ingredients deleted, along with an assessment of their full, partial, or no antimicrobial activity. So here, the gray bar is showing the control, again, of sterile distilled water. The red bars are showing the full and reduced versions for the onion eye salve. The blue is showing full and reduced versions for the leek eye salve. And the green is showing a variant of the recipe with both onion and leek deleted. So what to take away from this image? Deleting wine and garlic from both variants of the eye salve totally eliminates the bactericidal activity. So it would seem that the combination of wine, garlic, and the allium species are crucial for the full efficacy of Bald's eye salve. Also, when onion is used instead of leek, oxgall seems to be necessary. In both cases, brass is dispensable. So because of this combination of ingredients, we also looked at the 9-day activation period to see perhaps this recipe, there might be some methodology to it. Here, again, a control of sterile distilled water and the 2 variants of the isolve were added to Staph aureus growing in a wound biofilm model immediately after preparation, and the bactericidal activity was quantified. Those results are shown by the shaded bars. The experiment was repeated after the 9-day activation period, and that activity is shown by the open bars. You can clearly see the difference in activity over this 9-day waiting period between the shaded and the open bars in both variants of the isolve. So what I've just talked about is in vitro results against Staph aureus. Because it performed so well in these tests, it was decided that it should be tested in vivo against a resistant pathogen. Tissue from wound samples of mice infected with MRSA were submerged in a control of sterile saline, vancomycin, which is a clinical last-line antibiotic, and then the 2 variants of the isolve. Again, here, asterisks denote treatments which are significantly different from the control. You will note that the last-line antibiotic, vancomycin, produced no significant reduction in viable bacteria. However, both of the isolves did. So it would seem that Bald's isolve shows potential as an anti-staphylococcal agent, not only in vitro, but in vivo against a resistant pathogen. So what about against a non-bacterial parasite? So leishmaniasis, for those who might not be familiar with it, it's a protozoan parasite infection. It is caused by the bite of female sandflies. It is a major problem in warm parts of the world, and it's becoming an increasing problem with large-scale displacement of humans and people living in refugee camps. And what it does is it causes very unpleasant lesions which are then susceptible to infection by secondary bodies like Staph aureus and other species. So this work has been done by Lee Haynes of the Liverpool School of Tropical Medicine. She's pictured here in the slide taking a swab from a patient. And the other image that you see is a Giemsa stain slide of human macrophage cells. The gray area are red blood cells. The purple area is the macrophage. The large dark purple area is the nucleus, and then if you look closely, you can see dark purple specks. Those are the Leishmania parasites. And what they do is they evade human immunity by hiding within human macrophages. They rupture the cell and then invade other macrophage cells. Oh, a word of warning before the next slide. I know we are not all scientists here, so I'm about to show a somewhat graphic image of a patient with pretty severely infected leishmaniasis lesions. So if you find that disturbing, perhaps proceed with caution. But here we go. So you can see the immense pain and suffering that results from these lesions, particularly once Staph aureus or another bacteria begins an infection. And especially for people who have been displaced, people living in refugee camps without adequate access to medical care. And once these lesions heal, They often cause scarring, pretty significant scarring on the face, and that can lead to a social stigma. It can lead to bullying and depression, a loss of education if children then don't want to go to school. And in some parts of the world, for young girls, it can lead to a loss of marriage prospects. So if you can track this and you live in a part of the world with stable access to medical care, you could receive treatment by cryotherapy or thermotherapy. But typically, the drug most usually used is called pentastam, and this is injected either intramuscularly or intralesionally. And depending on the type of species, Leishmaniasis species causing the problem, the treatment can be quite painful, quite long, up to 18 months in duration for some people. And you can imagine this is quite a disheartening prospect for patients. Some people will refuse the injections and try and treat it themselves by burning the lesions with cigarettes or with fire, trying to cut the lesion, which clearly is not going to help the situation. So, what does this have to do with the eye salve? In the assay shown here, live parasites were exposed to dilutions of both versions of the eye salve, both onion and leek, over a 2-day period. After the 2-day period, a dye was added in order to measure metabolic activity, to measure if the parasites are alive or dead. When parasites are alive, the dye turns bright pink. When parasites are dead, the dye stays dark blue. And you can see where it says onion variant and leek variant, the dye is dark blue. It's clearly dead parasites. Now, this is quite an exciting result, but it's also a preliminary result. Right now, the eye salve is being tested on human macrophage cells in order to ascertain its effects on human cells. So it has to be determined that the eye salve is harmless to human cells, but maintains its toxicity against the parasites living within them. To summarize this evidence that I went through a little bit quickly, all of the ingredients are necessary for the eye salve to work, except for potentially the brass elements. Also necessary is this 9-day activation period. This idea of a combination of ingredients in pre-modern medicines is quite a key idea. Why might this be working? It could be that the antimicrobial activity of the individual ingredients on their own is combining to make the eye salve greater than the sum of its parts. It could be that bacteria is being attacked on multiple different fronts. It also could be that this 9-day activation period, perhaps a novel compound is being formed over this time. These are questions to be answered by future research, and that is where it's heading. Another key point, the recipe is highly reproducible, which is significant. As of right now, 6 people have made a couple of dozen batches of the eye salve using fresh ingredients each time, and all of them work equally well against Staph aureus. As I alluded to, current tests are being done on bald eye salve for its effects on human cells, and that will determine where it goes from there. Also, an analysis of how it's working. Why is it effective? Is there a novel compound being formed? Why this cocktail of ingredients? And switching gears a little bit, You'll recall from the beginning I related story of Thomas Gale and the Lily of Medicines. So this is a text that I produced the first edition of for my doctoral work, and it contains a wealth of pharmaceutical information. So from that text, we're doing something a little bit different. There is a chapter on weakness of sight, and within that chapter, there are different remedies for loss of eyesight, debilitating vision, problems related to old age. And so there's a recipe that, in translation from the Middle English, it says, "God has not given us a better recipe than this one. It makes even old men to read tiny letters without aid." And so we thought, that is quite the promise. And so we decided to test that one against macular degeneration. That is an ongoing test at the moment being run by Lisa Chakrabarty at the University of Nottingham, but we do hope to have some results for that by the summer. Another interesting ingredient from the Lily of Medicines is plantago. Now, this may be an unfamiliar herb, but it has quite a long and exalted history in medical history, being used by Greek authorities all the way up to the present day, even in ethno-veterinary practices. One scholar has termed it nature's band-aid because it's so effective at wound healing, insect stings, and also infection prevention, which is also part of the recovery process. So right now, this is in theoretical stage, but we hope to look at Plantago major, Plantago lanceolata for effects in wound healing and infection prevention. Also, you'll see on here a cold and flu preventative. This is a drink that I have made based on, inspired by Dr. Bernard of the Lily of Medicines. And it's something I've been taking myself for about 3 years. Anecdotal evidence is very strong. Of course, I'm not going to give you the recipe because I cannot say test this on yourself. Definitely not. And do not test Bald's eye salve. But that one, because of this particular cocktail of ingredients and the anecdotal evidence, I think we could make a strong case for this one. We're also going to be testing that later in the summer and maybe develop something from that. So of all of these ongoing, interesting, exciting projects, for the remainder of our time, I'd like to focus in on a recipe database and a pilot study we're going to do on data mining the Lily of Medicines. Just a little bit more about this text. There it is at the Bodleian Library. It is the only extant Middle English translation of Bernard of Gordon's Lilim Medicinae, which was originally written in Latin and completed in 1305. Then the Middle English translation came about a century later. Latest known printing that we know of happened in the very late 17th century. So you can see from that date span, it was a hugely significant text through the medieval period, through the Renaissance, and into the early modern period. And like I said, it contains a lot of very interesting pharmaceutical information that was probably the most significant aspect of it. So it makes a very good text to base this pilot study on. What I've done, the Lily contains around 6,000 references to particular ingredients, 360 very specific recipes, which represents over 110 disease conditions. And I put these into a database that we hope to analyze, looking for non-random co-occurrences of these ingredient cocktails. All of these recipes that I've talked to you about just now, bald's eye salve, the remedy for macular degeneration, Plantago, nettles, even the cold and flu remedy, they've all been hand-selected by researchers using a priori knowledge. It's quite time-consuming, and we're wondering if there might be a faster way to identify these cocktails of ingredients to inform laboratory testing and eliminate this kind of hunting and picking method through medieval texts. So that is what we're aiming for in the future, and that this pilot study has already commenced. And hopefully, again, summer's going to be a big time. We're looking to have some data out in the summer. And not only would such a project inform laboratory research, which has implications for new drug discovery, but it could go some way to answering some of the questions I posed at the beginning of this talk regarding the methodology of medieval medical practitioners. Often it's viewed as, when you see the long lists of ingredients contained in medieval texts, you think they were just trying anything they could without thought. But it would seem there is more methodology to these practices. Even in The Lily of Medicines, he says, if you don't know what to do, try this remedy, see what happens. But when you look at recipes like bald's eye salve, meticulously put together, even down to the amount of time it takes to produce this bactericidal activity. So we hope to explore that as well. This is a little bit of a whirlwind tour through 2 years of research. And I would like to close with just a few concluding thoughts on the great virtue of interdisciplinary and international collaboration. The arts and the sciences are often presented as these diametric opposites. But hopefully, in this talk, I've shown how much we need each other, and that when we work together, we can produce results that are greater than the sum of our individual parts. Clearly, not every collaboration is created equally, and it requires very specific traits to be part of such a well-integrated and unified team. Turning historical medical texts into new drug discovery requires very close working relationships with very different disciplines, excellent communication skills, open-mindedness, accessibility and discoverability of data, and knowledge exchange across such a wide variety of disciplines. In our team, we have parasitologists, medievalists, historians, climate historians, microbiologists, medicinal chemists, data scientists, mathematicians, spreading the disciplines and across countries. We might summarize that by saying a global problem requires a global mindset. And with further research in this spirit, it may be possible to show that the ancient biotics of yesterday have real implications for the antibiotics of today. And I know we're a little bit early, but that leaves lots of time for questions. Thank you so much for your attention. Very happy to discuss further. Yes.
So if bald's eye salve work well with some of these conditions, why do you think it fell into disuse?
Disuse in medical history.
Disuse, well, presumably at some point people stopped using it because even the woman that we're told about in the little story had to go look it up in an old book because the doctors at the time had forgotten about it.
Oh, yes, sorry. Thomas Gale's remedy, that was a different eye remedy to bald's eye salve. So And for those who might not have heard the question, she was asking, why did Ball's eye salve fall out of use if it was so effective? So Thomas Gale used a different eye remedy. But yeah, at some point, we did stop using this combination of ingredients to put on our infections on our eye. So there could be a couple of different answers to that. One is the view of the medieval period as a dark age. Other generations looking back and saying, no, we are far more advanced. There's nothing we can learn from that. That has been pretty permanent outlook for a long bit of history. Second, all bacteria will develop methods to the antibiotics, whatever form it is, that they're being exposed to. So they will develop resistance to them. So it's possible that Ball's eye salve and other treatments being constantly used, bacteria are constantly being exposed to that form of an antibiotic, that they developed resistance to it, so it didn't work. And we've not used it for all this time. Maybe that's one of the reasons why it's showing such efficacy. And if nothing else, perhaps something like it, now this is speculating far into the future, if it was to become something that was useful as an antibiotic, you know, eventually bacteria would become resistant to that. But it could buy some time. I mean, thinking about that statistic, which was put forth by the AMR review, Antimicrobial Review 2014, 700,000 people currently dying, up to 10 million estimated by 2050. I mean, it could buy some time, and then there could be a return to other antibiotics. So, and part of that is the antibiotic revolution. We live in very unique times with laboratory science, and although it is true that a lot of our compounds that we would see as pharmaceuticals are derived from natural sources, it often doesn't come to us in that way. So there is a connection there of using nature to find new antibiotics. So did that answer? Yeah. Yes?
You said that wine is part of it. What kind of wine do you use?
Yes.
I'm sure I'm not the first one to test.
No, that's a very good question. Actually, I brought the paper with me because I want to get it exactly right. It's actually where the wine was sourced. It was an English white wine from an area of the country where an old English monastery used to own the vineyard there. So it was as close as we could get. Yes, here we go. Organic English white wine produced by a vineyard situated 7 miles from the village of Panborough in Somerset. A charter of AD 956 tells us that the Anglo-Saxon monastery of St. Mary's Abbey in Glastonbury held land rights to a vineyard at Pemborough. So we also, with the honey as well, because obviously this is not an exercise in trying to replicate the medieval past, which we would argue is probably somewhat futile, because we cannot replicate those conditions whatsoever. Our purposes are totally different. But we did try and get close to ingredients like the wine. The honey was sourced from, I believe she's in Surrey, a lady in Surrey who had recreated a medieval garden with medicinal herbs and plants, and bees nearby were pollinating those plants. So that's where we obtained the honey from, and just for some continuity. Yes?
I have two questions. One was, in your studies, did you find that the Ball's formula was bactericidal or bacteriostatic? Those two. And the other one is, I'm kind of mystified by the 9-day shelf life, or the actually I'd like to know the shelf life. Once it reached the 9-day, was it potent until 9 days, which was probably just serendipitously discovered, right?
Mm-hmm. Trial and error, yeah.
Yeah, and how long was it good for thereafter?
Yes. So, I'm not a PhD microbiologist, but I'm going to answer this. I will recommend that you look to the paper as well, and anything I don't answer, contact Freya Harrison or Steve Diggle, whose email addresses are on the paper, and they'll be happy to answer it. So the question, for those who didn't hear it, was Ball's eye solve bactericidal or bacteriostatic? And the answer that's coming to me from the paper is bactericidal. And in the results as well, it was found the word design is actually been used in reference to this recipe, in that it treats the infection that it was designed to treat, almost in a targeted way. And the isolve has been tested against other pathogens such as K. pneumoniae and E. coli and Enterobacter and things like that. And the activity is different. So it's not like dumping bleach on something where it just kills everything. It seems to be specifically targeted to what it was made to treat, which is very interesting and worthy of more research, and that is ongoing. And then the other question was the isolve good for the entire 9-day period? What was its activity through that period, and how long does it stay good? That is actually being looked at the moment. There have been some old samples of the isolve that are being tested. I was actually just speaking to the microbiologist who performed this work, and she has some old samples that they're testing. So I'm not sure about the terminal dates, how long it stays good. I believe it has been tested, but I can't give, like, it's bad after 30 days or it becomes toxic after this period.
They wouldn't have any refrigeration, obviously, so this would be basically at room temperature.
At room temperature, and that was a consideration in running this test. There were a lot of decisions that had to be made, and it was decided to keep the isolve in the refrigerator because in the building where the lab was, the heat comes on and off, and it was decided that fluctuations in temperature like that would be more unrealistic than keeping it at a steady temperature. Because often in other medieval recipes, actually the one with macular degeneration, one of the instructions says, make it as hot as a hen's nest when she's sitting on the nest. Like, what temperature would that be? So, find a hen. Yeah, or, so that is during the winter. If it's being made during the summer, the recipe is to sit in a cool, dark shadow. So it's what they had in their environments. But they're looking for a steady temperature. With the hen, steady temperature, dark shadow, steady temperature. So that's why we chose refrigeration. Yes?
You mentioned that one of the things that you've discovered is that the Ball's isolve recipe, for instance, is highly reproducible. You can make the same thing again and again. Is there any intent to try making the recipe with modern wine and honey, since those two ingredients seem to be somewhat limited in availability?
Well, we could say it was made with modern wine and honey because we don't really have access. Like, all of these ingredients are modern in the sense that, sorry, for those who didn't hear the question, it was, has there been any attempt to perhaps change the wine and honey other than the ones that we used in the original test?
Yes.
Yeah, okay. No, it's, well, with the 6 who've reproduced it, I'm not entirely sure if they used the exact same sources, although I think they did because they were trying to replicate it exactly. I know that in the recipe that I made with the cold and flu preventative, I've changed up the honeys multiple times, and we still need to test this. But I will say that Manuka honey would be worth trialing in one of these because it has the highest antimicrobial content of any honey, and it's what I use in my recipe. Maybe it's a placebo effect because I really believe in it, but that waits to be seen. Yeah, because often medieval recipes, even with vinegar and with wine, they'll say, make it a weak wine, make it a strong vinegar. And sometimes they don't give a lot of guidance to what that means. Obviously, with a weak wine, you would think perhaps it's newly made or they're diluting it with water or some other ingredients. But yeah, did that answer?
To a certain extent, but one of the things that I was more curious about is if these recipes prove to be efficacious,
Mm-hmm.
then in order to ramp up production, you have to be able to source ingredients on a mass scale. And it seems to me that a small vineyard or
Yeah.
a local beekeeper aren't going to serve for mass production.
No. In that case, we would have to find a different source. And I would say Manuka is one of the best that we could do. But no, that tiny vineyard and the poor beekeeper, no. For worldwide production. But of course we're not there yet. So that was mainly done for the sake of the pilot study and to keep it as close to the Anglo-Saxon source as possible. And also because no one expected it to be so effective. It was what we could call a Friday afternoon project. Everybody did this in their spare time, kind of still doing it that way. And it was really quite shocking when these results came back. And right now, the recipe, we're looking into the mechanism of action and that kind of thing, but its effects on human cells need to be looked at first before we look at anything else. But yeah.
Thank you.
Thank you. Yes.
Someone online asks if your team had any thoughts about substituting the ox gall.
No. Did they mention what they thought it might be substituted
No,
with?
they mentioned the possible objectives, which could be for vegans or those with allergies.
Ah, right. The thought with the ox gall, because, here I'll show you. That is a good point. So, in this graph, you see with the leak version of the iSolve, ox gall is not necessary. With the onion version, there's only partial activity when it's omitted. So it's an ingredient sort of like brass that could be dispensable. The iSolve is not showing its full activity with that ingredient present. So in the future, it could be something to look at. I'm not sure what we would substitute it with, because it's bovine bile. It can, like bile can act as a surfactant. I'll think about it. Yes?
Are you getting any interesting responses from the microbiology research community that you can mention?
Yes, actually. So the question was, have there been any interesting responses from the microbiologist community? And yes, actually, in 2015, when this data was announced and published, it actually attracted worldwide attention. And the two leads of the project, Christina Lee, who's the Anglo-Saxonist, who translated the recipe, and Freya Harrison, who led the microbiology work, they did a lot of interviews with worldwide news outlets, and the paper has quite a high impact rating, and it kind of went all over the world for a time. And responses from microbiologists, unless there's something I don't know about, they've been very positive. And I think people are quite inspired, not just by this research, but by lots of research going on with nutraceuticals and looking to the past for inspiration. There's even a team in Sweden, I believe, that have just produced mead, Viking mead, which is highly antibacterial, and they produced it for sale, so you can buy it if you like. And for the person with the honey question, they did use honey in it. It'd be interesting to note what honey they use, because I'm not sure. And I just saw that data last night. So I think we're right on the verge, perhaps, of something. But yeah, responses have been very positive, and I think people are quite excited about this research and the potential it has. Of course, this is very preliminary. It has to be tested on human cells before any more progress can be made. But, yeah.
Do we have any further questions for Erin? And I do have to say that in New Zealand we use Manuka honey on kind of everything.
when you walked in, I thought Manuka, New Zealand. But yes, thank you all very much.
Thank you. Please join me.
This has been a presentation of the Library of Congress. Visit us at loc.gov. This has been a presentation of the Library of Congress. Visit us at loc.gov.